In this lecture, we're going to be talking about myofilaments. If you'll remember from last lecture, it's the myofilaments, the thick and thin filaments, that work at the molecular level to cause contraction. And the myofilaments will make up the sarcomeres, and the repeating units of the sarcomeres will make up the myofibrils, and the myofibrils will make up the muscle fibers, the cells of the muscle tissue. There were two kinds of myofilaments. There are thick filaments, and the thick filaments are composed of a protein called myosin. And what will be key here is that myosin will bind to actin and will form cross bridges with actin. It will pull on actin to cause contraction. And the molecule that it will use to provide the energy for that contraction is ATP. And ATP is the cellular energy source for all cells. And then we have the thin filaments. The thin filaments are primarily composed of actin, and it will be myosin binding to actin that will cause contraction. But the thin filaments also have two other key proteins associated with it, the tropomyosin and troponin. And these are regulatory proteins that, under the influence of calcium, will ultimately turn contraction on and off. If we look now, let's take a look at that structure. So here are the myofilaments. Notice you can see how these myosin heads will be able to bind to actin and pull to cause contraction. If we look at the thick filament is composed of the myosin molecules. Notice that it has this flexible hinge region. This is it will be the head of the myosin that will bind to the actin and pull to cause contraction. Here we have the thin filament. It's primarily composed of actin. Actin are these kind of little blue balls. Notice that on the actin, if we blow up that actin molecule, the actin has what are called active sites for myosin. And it's on these active sites on myosin, it's on the active sites on actin that the myosin heads will bind and pull to cause contraction. Notice that here is troponin, these little yellow balls, and here is tropomyosin. At rest, tropomyosin is held in place by troponin and covers the active sites of actin. And so myosin cannot bind and the muscle is relaxed. What will happen is that we'll have an electrical impulse that will release calcium that will bind to troponin. It will move tropomyosin, and now those active sites on actin will be exposed, and myosin can bind and cause contraction. Thank you.